The overall goal of this procedure is to derive DNA strand displacement gates from bacterial plasmids.
The main advantage of this technique is that it utilizes bacterial plasmids as a highly pure source to generate robust DNA gates. Demonstrating this procedure will be my colleague Sundipta and myself.
To begin, mass produce and then isolate plasmids containing DNA gates by using DNA isolation kits as described in the accompanying text protocol and in accordance with the manufacturer's instructions. Following elution, measure the concentration of plasmid DNA using standard techniques. Then digest the DNA by adding 4 units of the restriction enzyme PVU2HF for each milligram of the plasmid. Next, add 2 equivalent volumes of ice-cold absolute ethanol to the sample. Incubate the mixture at -80 degrees Celsius for at least 1 hour to precipitate the DNA. Then, pellet the precipitated DNA by centrifuging the sample at 10,000 to 14,000 times G and 0 degrees Celsius for 30 minutes. Remove the supernatant and add 1,000 microliters of room temperature 95% ethanol to the sample. Then invert the sample 10 to 15 times. Next, centrifuge the sample at 10,000 to 14,000 times G and 4 degrees Celsius for 10 minutes. When finished, remove the supernatant and air dry the DNA on a bench for 10 to 20 minutes.